Hello everyone, welcome to today's live broadcast, Innovative Quantitative Imaging Techniques and Their Utility in Advancing Neurobiology Research. I am Marjorie Torres of LabRoots and I'll be your moderator for today's event. We are excited to bring you this educational webcast presented by LabRoots and sponsored by GIBCO, part of Thermo Fisher Scientific. GIBCO media supplement cells and cell culture reagents are designed to deliver reproducibility and performance for results you can count on every day. For more information, please visit www.thermofisher.com/GIBCO. We have a few important announcements before we begin. This webcast is designed to be interactive and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right-hand of your presentation window or submit your problem through the green Q&A button lower left. I would now like to introduce today's speakers, Dr. Daniel Beacham, Dr. Nicholas Radio, and Michael Derr. Dr. Dan Beacham is a senior staff scientist in the Discovery Biology Group at the Molecular Probes campus of Thermo Fisher Scientific in Eugene, Oregon. There he developed cell-based imaging, high-throughput screening, and biochemical tools for drug discovery in basic sciences research. Dr. Nicholas Radio is the manager of cellular imaging and analysis for Thermo Fisher Scientific in Pittsburgh, Pennsylvania. He specializes in high-content analysis with expertise in drug discovery and in vitro toxicity, with emphasis on development of an in vitro cardiac, neuronal, and hepatic toxicological assays. Michael Derr earned his Master's of Science from Oregon Health and Science University. Michael's research is focused on development and commercialization of next-generation culture systems for pluripotent stem cell culture, differentiation, and neuronal cell culture at Thermo Fisher Scientific on the cell biology team based in Frederick, Maryland. I will now turn it over to Hunter Tuck, Product Manager of Thermo Fisher.
Hi, thank you. Yes, this is Hunter Tuck. I'm the product manager for our neurobiology portfolio of products here at Thermo Fisher, and we're very excited to have this second webinar today focused on the tools and techniques to advance your neurobiology research. This is a second in a series of webinars that we'll be offering throughout the year focused on solutions in neurobiology research. If you haven't already, I would definitely recommend checking out the first one that we recorded back in May, titled Improved Differentiation of Stem Cell-Derived Neurons in Downstream Applications Through the Reduction of Progenitor Proliferation with CultureOne Supplement, which was presented by our own Dr. David Cunninger of R&D and our cell biology business. That webinar specifically featured a new product called CultureOne Supplement that we debuted last November. And for more information on that product, please visit our website, thermofisher.com/cultureone, or simply Google CultureOne. On that site, you can learn all about the new innovative products that will hopefully help you improve your downstream research, some of the techniques of which we will talk about today. And you can also check out our Neurobiology Protocol Handbook, which features all of our solutions across the neurobiology research workflow. So without further ado, I'll turn it over to our first speaker, Dr. Dan Beacham, who is a senior staff scientist in the Discovery Biology Group. Thank you, and take it away, Dan.
Great. Thanks so much, Hunter. And thanks so much for everyone who has tuned in to listen to our broadcast today. I'm going to get to talk about an extension of the work that Hunter mentioned with the GIBCO team from the webinar that went up in May. A collaboration with David Cunninger and his team out there in Frederick, Maryland, working on new media formulations to enhance and speed the differentiation of neural stem cells for functional assays. And so the functional assays are really what we're going to take the deep dive to in this part of the talk. So here, I'll just give a very brief overview of the content we'll be looking at. A couple of slides looking at why we use fluorescent labels in this kind of functional assay. Then I'll go into the meat of the talk, describing how we used calcium indicators and a cell proliferation assay to monitor the progress of the cell differentiation in CultureOne medium. And then I'll introduce a couple of additional assays that we've developed or developed further for interrogating excitable cells. Of course, here at the Probes campus, we still are very much focused on fluorescent methodologies. And so, of course, what we're going to be looking at in our graphical and tabular format are signals from cells that we're going to record as an output of their activity. So in the case of neural cells, we're going to be very interested in activity linked to action potentials, and our indicator here is going to be around calcium. We use fluorescence, of course, to enhance the contrast of an otherwise relatively low information content and otherwise invisible signal. On the left, you can see some cardiac myocytes on a still image just kind of sitting there. And on the right, what you can see are these cells when they're loaded with a calcium indicator dye. And in the bottom left, behind the arrow, you can see the calcium oscillations placed from the cells, from a signal, from a reference in a region of interest over one of them that shows the flux of calcium in and out of the cells during their rhythmic contractual beating. So that's, of course, an example for cardiomyocytes. We're going to use the same methodology to interrogate the activity now of neural stem cell derived excitable cells in the second part of the talk. So jumping right into it, as Hunter mentioned, you got an introduction to the CultureOne media product and its use from David Cunninger back in May. And I just kind of wanted to highlight, again, what the aims of the study that we're going to look at will be. Our intention in this collaboration was to prove some of the hypotheses we'd put out there in looking at the cells after they were differentiated. So this product really is intended to cut down on the number of contaminating non-differentiated cells that run amok in traditional NSC cultures, and can greatly blunt the responses in the downstream assays like these functional readouts. I don't have any content to share this time on its effect in primary neurons, as shown below there, but watch this space for some exciting new data as we develop further this space. So I just wanted to state the problem with these cultures. Typically they run some 3 weeks to get to the end point where we want to sort of ask the questions of the cells, either from a patient-derived sample or from a progenitor cell line about their activity. And these what's meant to show, what's shown in red here are some of the problems that we're seeing in this field as it grows. The first and foremost being that they're no longer a pure population of neurons, or rather of cells, period. They're a mixed population of progenitor cells and cells that are either in different states of division or have not yet committed to a differentiation pathway. So this is meant to show what the issue is. Some staining in the lower right, showing this mixture of progenitor cells as indicated by the red SOX1 staining, and then differentiated neurons by MAP2 staining. Next slide shows a little bit more closely, and then more importantly, what the effect of CultureOne is. We see a dramatic reduction in the SOX-positive cells, and then a further enhancement of the MAP2 neural cell marker. And then across the top, you can see, again, one of the issues that we encounter in culturing cells in the absence of CultureOne for these 2- and 3-week time periods that are required to get cells to a desirable endpoint. So the study that I'm going to outline for you today is, again, designed to interrogate the cells at several time points for functional activity. So The design of the experiment here is to plate the cells just very simply with and without the reagent, CultureOne, at a fixed density. And then each week I'm going to do 2 tests. One test of the excitability. I'm going to index the excitability by the cell's response to potassium depolarization. I'll have some more detail on that. And then I also want to look at the cell numbers that are in each well to ask, what the status of the proliferation is and relative numbers of cells, and more importantly, something that I can index the calcium responses to. I'm doing this on 2 platforms, our own Thermo Fisher VarioScan flash plate reader just for the endpoint CyQUANT direct cell proliferation assays, and then the Hamamatsu FDSS 6000 high throughput plate reader, which is a kinetic imaging plate reader with fluidics capabilities that I'll go into a bit more. And again, these 2 questions we'll be asking are the cells still dividing, and are they excitable? This slide is a sort of a close-up of a snapshot of some HTS data, some high-throughput screening data as you would see it on the screen. These are actually averages of 4 wells over many time points, and what you can see at the base of that curve is where the injection of the stimulus is. If I inject a control solution with no additional potassium chloride, I get a sort of a little bump, a stimulus artifact, we'd call that. But if we add 30 millimolar potassium to the cells, this depolarizes the membranes and recruits the activity of voltage-gated calcium channels. We know that voltage-gated calcium channels are an important hallmark of neuronal, they're one of the more important proteins related to neuronal function that gate transmitter release and a number of other signaling and action potential propagation properties of differentiated neural cells, and they're largely absent from the progenitor cells that we'll be starting with here. So this is intended to show the kind of data that we'll be getting, and the pictures on the left just are some microscopy images that I've taken to show you. Again, this is what you would see in the microscope, and then on the right is what the photomultiplier sees in the high throughput instrument. So now we're going to take a closer look at this data so that we don't have to look at curves and images anymore. I just want to show you nice bar graphs so we can highlight the reproducibility of the study, as well as get a closer look at the comparative amounts of signal. So all I've done, the manipulation of this data is to export the peak and then the time period of 2 seconds on either side of it from each well, and then place those into a spreadsheet and average them with the other 3 samples that were, in this case I did 8 wells per sample, and then so average them against each other to give the size of the response in the various conditions. I'll point out the y-axis here. This is a measurement in high throughput called delta F over F. That's fold increase in signal. So really that's just a running average of the starting signal at 1.0, and then following the stimulus, what the relative change is in time as the signal progresses. So the first bit of data that we got here, we'll be looking at data from week 1, week 2, and week 3. So here we are on day 7, and I set this up so that I could look at a few things. I was showing you maximum responses to a 30 millimolar potassium chloride stimulus. Here I've shown a bit more detail. I actually stimulated the cells with control over there with 0 potassium, and then increasing doses of 5, 10, or 5, 15, 20, and 30 millimolar to kind of ask, number one, what is the maximum response possible, but do we see responses from the cells at the lower doses as well? And we'll largely just pay attention to the maximum, but you'll be able to look at these lower dose signals as well throughout the study. As I mentioned, part 2 of the study is to look at the cell numbers. So after 1 week, just looking in a white light microscope on an EVOS, I think it's one of our cell culture microscopes, I can see already differences in just the morphology of the cells, and also in the cell numbers as the cells in the absence of CultureOne medium are proliferating and kind of encroaching in the middle of the field, and the cell numbers in CultureOne have stayed the same but are elaborating long processes. So the assay that I'm going to use to interrogate the cell number is a simple one, one we like a lot because it doesn't rely on the presence of ATP inside of the cell, is not sensitive to contact inhibition, and importantly, these are cells with long membranes and lots of membranes and long processes. So what we do with this means of following proliferation is count the number of nuclei. It's very simple. We have a very bright green nuclear stain that we apply to the cells in the presence of an extracellular quencher. Cells that are compromised, that have been either killed by a drug or are just simply not there, the signal from the nucleus will be quenched because the dye can enter these damaged cells, and then living cells are left behind. So this is a life-positive signal that reliably records the number of living cells in a culture. That's illustrated in the cartoon, and already what we see on day 7 is that there are about 6-fold more cells in the absence of CultureOne media than there were in the presence, and so cell numbers have stayed the same relative to the day 0 data in the presence of CultureOne. And the next few slides are just going to now review days 14 and then days 21. So here we are on day 14. Now we're starting to see that the real problem with growing cells in these conditions, they are stacking on top of each other and just overgrowing the plate. And then the cells in the presence of CultureOne, again, no change in the density, but much more in the way of processes that we saw the staining for earlier that are going to be positive for MAP2. And now, so here summarized on this slide is the CultureOne quantitative data, the CyQUANT and Flow4 quantitative data, looking at the cell numbers and the relative responses of the cells on day 14. So despite the fact that there are 20-fold more cells in the control condition, we have smaller responses in the Flow4 assay. And skipping on now to day 21, cells again showing clumping and this rather sort of a difficult to image phenotype. We can now look at the cells again with these two assays, and now we're really seeing a big uptick in the CultureOne derived cells. So without any need for normalization, what we're seeing is that there's about twice the signal coming from 1/20th of the cells in the calcium assay, and that speaks quite strongly of a greatly increased expression of calcium on those cells, calcium channels on those cells, and the responsivity to the depolarizing stimulus. This last slide summarizes all of this data together from day 4, day 7, day 14, and 21. Again, highlighting the endpoint of the study, in particular the day 21 difference between the cells with and without CultureOne in response to 30 millimolar stimulus. So now, in the end of my talk here, I will go ahead and introduce a couple of other assays that you can probably expect to see from us in this place. We'd like to start interrogating these cells with other readouts besides calcium to ask dive a little bit more deeply into asking what other channels that they might be expressing, and also just introduce these assays as important tools for any neuro or clonal ion channel type research that may be going on in your laboratory. So the two assays I'll introduce here are one, a potassium ion channel assay called Fluxor, and we've recently re-released this as a greatly improved and larger signal-to-noise assay. We've also released a red form for working with GFP, and then also a very fast and high-fidelity membrane potential dye called FlowVolt, which is used to record action potentials and other fast events at the membrane in an imaging mode. I'll introduce the problem here by stating, the way you traditionally measure channels over there on the far left by conventional patch clamp. is, one cell at a time, and hours in a day, and it takes weeks to get, good N numbers and dose responses and so forth. The advent of automated patch clamp has ameliorated this somewhat. It is extremely expensive, but it affords some throughput to otherwise very difficult or intractable targets. And then on the far right, that number of targets diminishes even further, but has much higher throughput in the HTS high throughput space, where we can use fluorescent kinetic assays to report the activity of cells. So what I want to show is in this first panel of assays under the Fluxor category here, we've opened up a whole new arm of the chanome, the ion channel compendium of genes expressed by mammalian cells, by introducing the Fluxor assay, and opened this now to the high throughput space. We did this by using an approach with a surrogate ion. Potassium is actually very difficult to measure directly, and in fact the potassium changes inside of a cell are very small. So even if you could measure them, it'd be very hard to index the changes in potassium to the number of channels that were open. So what we did instead is develop a dye that would report the activity of thallium inside of cells. Thallium selectively enters open potassium channels, and in fact passes through, is transported through potassium selective transporters. And then that gives us a bystander kind of an ion that shows the relative number of open channels in any given cell population by the addition of thallium. So it's very easy to make a thallium-sensitive dye that will do this here with our chemists. And so we developed this assay really to interrogate the activity of potassium ion channels and transporters, and even the activity of G-protein coupled receptors that couple to them, as shown on the right. Our first generation of this assay was launched in 2009, and some years later, a competitor assay appeared. You can see there in the middle set of graphs in the top left that had a slightly higher signal to noise, and so we went back to the bench and produced an even further improved signal here with a dye that's much more sensitive, and also gave us the luxury of producing an assay that retains all the pharmacological fidelity you see in the lower right against a common drug that E4031 used against the hERG cardiac potassium channel, but also gives us the opportunity to do the assay in no wash. This next slide just simply shows another target, a mixture of ion channels, the KV 7.2, 7.3 ion channels, interrogated with either an antagonist, a partial antagonist that inhibits the current and knocks the signal down, or an agonist that opens the channels and causes an increase in the signal. So this assay isn't just for looking at blockers. This slide shows that the new Fluxor 2 assay can be configured, is very, very flexible, and can be configured in one of three ways. Either the traditional two-wash assay, where the dye is added and removed, and then you can replace with assay buffer and have conditions very controlled. You can also do the assay in the absence of media simply by removing the media and replacing with dye containing buffer that's very dim at basal rest, so a basal signal that doesn't interfere. Or you can do this in a more desirable and high throughput manner, where you simply add the dye as a 2x formulation to the cells, do the assay in complete media, and get the data. And you can see in the bottom there that removing those wash steps increases the Z prime, because there are fewer steps that you have to manipulate the cells. A companion to this new tool is the Fluxor Red assay. Really should be called the Fluxor Orange assay. This is the Tritzy dye, exciting at 550. But we developed this to address questions from researchers largely in this space, working with neural stem cells, where GFP is often on board as an indicator. So the presence of GFP can also be a confounding factor, and the need for discovery screens outside of the green spectrum has been a known demand to us for some time. We also wanted to make sure that that researchers could do compound library screens as a secondary or orthogonal with a different dye to get around possible autofluorescence issues in the libraries. So just to briefly summarize the Fluxor kits, we have a new Fluxor dye, Fluxor 2 green, with a greatly improved signal to noise, great flexibility in its need for wash or not. And then I'd say our Fluxor red assay is very similar to the first generation Fluxor green assay. It does require a wash step, and it gives about the same signal to noise as the original. The next slide is an introduction to a fast membrane potential dye that we launched that we've named FlowVolt. And this is shown in comparison with one of our frontline fast dyes, diate ANEPs, in a patch clamp experiment where we've clamped the membrane potential and then stepped it to a series of depolarizing values and stepped it back and recorded the fluorescence from the cells. And what that has allowed us to do is load cells, like in this case a neural stem cell derived sample, and place a region of interest over, say, a cell that you might be interested in, and monitor the voltage excursions in real time with very fast refresh rates and resolve action potentials or clusters of action potentials on these cells. This is actually quite undersampled at, I think, 10 millisecond intervals, but we've now got reports from researchers that you can sample in a sub-millisecond range and actually record action potentials from the base to the peak. So membrane, allowing the measurement of membrane voltage as well as the flux of particular ions like calcium or potassium. I'll conclude here just with a note about the three things I've talked about. So CultureOne Media Supplement and our studies there do show an enhanced, not just staining, as you'll see from Dr. Radio and Michael Derr in the talks to come, but also enhanced function, as shown by our calcium assay. And then some new generation ion indicator assays that we've developed in the high throughput and discovery space to assist scientists working with excitable cells. So with that, I think we're taking questions at the end. So I'm going to turn it over to Dr. Nick Radio, who is Manager of Cellular Imaging and Analysis, and is going to introduce a different kind of microplate-based assay in high content screening.
Great, thank you so much, Dan. So what I'm going to be talking about today is the need for high content technology to evaluate developmental neurotoxicity. This is a subject that's pretty near and dear to my heart. It's a way of evaluating different developmental disorders, things like autism and ADHD. The backstory is that the traditional methods to evaluate developmental neurotoxicity, they take a lot of resources because they're animal-based, and because of that, it can take literally years to get risk data that is necessary for safety assessments. So it's resource prohibitive to attempt to use these traditional models, given the fact that there are literally thousands of compounds that are out there that are used that we don't have adequate safety data on. So the challenge becomes, how do we develop more efficient screening assays to give us some safety data on these compounds that are out there? So what we are going to be talking about is moving the developmental testing into a cellular model, an in vitro model. And that's based on the premise that normal development of the brain is dependent upon several critical cellular processes. So these include things like cellular proliferation, differentiation into mature neurons, migration, neurite outgrowth that I'll be focusing on here today, synaptogenesis, myelination, and finally programmed cell death. And again, it's based on the principle that neuronal cell cultures in vitro are able to recapitulate these processes that we see in vivo. So neurite outgrowth has received a lot of attention in the DNT space because it's one of the principal characteristics of a neuron that is differentiated. This slide here shows two different models. On the left-hand side, those are primary cerebellar granular cells that are exposed to serum to allow them to differentiate. And on the right-hand side, these are cells called NS1 cells. It's a PC12 subclone. And at the top there, you can see the cells undifferentiated. After exposure to nerve growth factor, you'll differentiate the neurons, and you can see the neurite outgrowth as shown in the bottom right. So even in vitro, it is difficult to identify and detect the neurite outgrowth. It typically would involve trained technicians to sit down and manually trace each individual neurite, which is a pretty time-consuming process, and it is prone to scientist variability when they're doing that type of manual methodology. So with this technology, we're utilizing the high-content screening to conduct the analysis. And the timeline, it's as efficient as you're going to get. The NS1 cells, they take about 4 days to differentiate following exposure to the nerve growth factor. The immunostaining of the cells, that takes about 3 hours from fixation, permeabilization, incubation with the primary and secondary antibodies, and then finally you're ready to run the analysis in an automated fashion using high-content technology. And just to give you an idea, so to look at a 96-well plate, an entire 96-well plate with 3 images acquired, and in each of those images we have about 100 cells that are analyzed. If I take 96 wells, multiply that by the 3 images, 100 cell density, and then 8 endpoints, things like neurite outgrowth, branching, cell size, we can generate 230,000 data points per plate, and that's done automatically within about 10 minutes. To give you an idea, if we did this manually, it would take closer to 100 hours to do the same type of research. So it's significantly more efficient. What I'm going to go into now are the different optimization work that we did to maximize the sensitivity of the assay. So this is just some preliminary work. What you see here is this is a dose response curve of the nerve growth factor inducing agent. It's in nanograms per milliliter. And you can see this is looking at neurite outgrowth total length. So anything greater than 10 nanograms, we see a significant increase in the maximal neurite outgrowth length. Just to be safe, we chose a saturating concentration of 100 nanograms per milliliter. And that way, we repeatedly were able to show nice, robust neurite outgrowth in the PC12 cells. Some other things just to show you. So in addition to the neurite outgrowth, you can see neurite count, so the number of neurites coming off of the cell body, the cell body area, so of the soma, and then finally the total branch points. And you can see within each of these other measurements, at 100 nanograms, we were saturating the cells to induce the differentiation into the neurons. Okay, and then finally, I wanted to just show, as far as the time courses and some different compounds that we did look at for control purposes. In the upper left-hand corner, what we're looking at here is with and without nerve growth factor at that saturating dose of 100 nanograms per milliliter. So what you can see is that at a period of 96 hours, we see a significant increase in total neurite length, especially relative to the untreated control. So in normal healthy cells, we would expect to see approximately 80 microns of total neurite length in the differentiated neurons. In the upper right-hand quadrant, so this is showing DMSO vehicle effect. DMSO was the vehicle that all the compounds that we evaluated were suspended in, and we wanted to know, does the DMSO vehicle itself cause any effect? And actually it does. You can see as we increase the concentration of DMSO, we do see a dose-dependent decrease in total neurite length. So to be safe, we chose a final concentration of 0.1%, which was, it induced a non-significant change in total neurite length. And then last thing is to ensure our biology was working, we wanted to identify a pharmacological control that we could use to show that the expected neurite outgrowth inhibition was occurring. as a way to biologically test the conditions in the plate. So the compound that we selected after evaluating a few is a protein kinase C inhibitor called bis-indole-maleimide 1, or BIS-1. And what's really nice about bis-indole-maleimide is here in green, this is total outgrowth, and we see a very nice concentration-dependent decrease in neurite outgrowth at concentrations that had no effect on viability, indicating that it was a specific effect to the neurite outgrowth biology in comparison to a more generalized effect. So we used the bis-indole-maleimide at a concentration of 3 micromolars to actually show that we were inhibiting neurite outgrowth as expected with this control. And then finally, we wanted to evaluate whether or not we were seeing any plate layout effects. In other words, were cells that were seated on the exterior wells on our plate, were they any different than biologies occurring in the interior wells? So when I say interior wells, I'm talking about rows B through G and columns 2 through 11. And you can see when we treat the cells in the same conditions, the exterior wells versus the interior, there's no difference in the neurite outgrowth length. So we felt comfortable in using the entire plate for our 96-well screen. So long-winded answer, but this was how we optimized this assay. We took 2,000 of our NS1 cells. We incubated them with and without 100 nanograms of nerve growth factor per milliliter within the 96-well format. We allowed the cells 2 hours to adhere to the bottom of the plate, at which point they were exposed to the compound of interest that we wanted the safety data on. We chose 96 hours as our time interval based upon the maximum neurite outgrowth in control cells, at which point we fixed and stained the cells. That took about 3 hours, and then conducted the high content analysis within the system. In parallel, we also used viability assessment, and that was more so that we could interpret, was this something specific to the neuron? Was it a neurotoxic effect, or was it more of a generalized cytotoxic effect? So here I just have a plate map, and what you can see here is in column 1, we have our control wells. Just quickly, so A1, B1, and C1 was without nerve growth factor. That was our untreated population that served as our negative control. D1, E1, and F1 was plus NGF, so those were the healthy positive control cells where we would expect to see maximum neurite outgrowth. And then finally, G1, H1, that was the bis-indole-maleimide treated cells that acted as our pharmacological control. Over the map of the rest of the 96-well plate was devoted to our dose response curve. We selected a relatively broad concentration range, so 1 nanomolar all the way up to 100 micromolar. It was done in a semi-logarithmic increase, and we chose that broad concentration range because for the majority of these compounds that we were interested in, we didn't really know at what concentration they would be effective, thus the broad concentration range. All right, so what I wanted to share was some of the different patterns of effects that we observed in looking at these different environmental compounds. So diphenylhydramine, that is a compound that is classically thought to be without toxicity for developmental neurotoxicity. It's the active ingredient in Benadryl, so it's an antihistamine. And what you can see here is that concentrations from 1 nanomolar concentration all the way to 100 micromolar, there was no change. And what you see on the graph in green, that is total neurite length. So we can see all the way up to 100 micromolar, there is no change. And in red, that's the viability. So we're not losing any cells. We're also not seeing any change to total neurite length at any concentration tested. Conversely, we saw non-selective effects, like you see here with cadmium, which of course this is a heavy metal. And you can see, starting at concentrations of about 1 micromolar and up, we see a dose-dependent decrease, where both the viability decreases as well as the neurite outgrowth. And we would interpret this as a non-selective effect. meaning at concentrations that reduce neurite outgrowth, they also cause a generalized cytotoxic effect. And then finally, the compounds that were of most interest are shown here on the right. These are compounds that inhibit neurite outgrowth at concentrations where we do not see any change in cytotoxicity. And here you can see at concentrations very low, in the nanomolar range, we see this dose-dependent decrease in transretinoic acid. Transretinoic acid is the active ingredient in Accutane. It's a known developmental neurotoxicant, so it was reassuring that we were able to see this positive effect. And just to show that we can actually take this and scale it for a higher throughput, this was evaluating 320 different compounds. We evaluated them at a single dose response, and you can see that it was able to detect compounds that showed a significant decrease in both total neurite length as well as viability. So the proof here is that we can take this type of screen and efficiently run it through so that within a month we have dose response data on the entire test set. To give you a comparison, to do the same type of assay in your traditional rodent model would take 480 years, which obviously is time and resource prohibitive. So finally, I wanted to move along and just quickly discuss some secondary measurements that are of interest. The first thing that I wanted to show is this is just a cell model comparison where the dotted line here, we're now looking at cerebellar granular cells relative to NS1 cells. And the reason why I wanted to show this is that it does show differences in sensitivities to different model systems. So CGCs, these are primary cerebellar granular cells, and a lot of scientists would say that these are more faithfully recapitulated in the in vivo system. So here with methylmercury, this is a known DNT, and you can see that both compounds, or both cell models rather, show the inhibition, but we do see a higher sensitivity in the NS1 cells. It's even more drastic here with transretinoic acid. So as shown before, NS1 cells, we see this significant decrease rather, in the neurite outgrowth in the NS1 cells. With the CGCs across all concentrations, we hardly see any change. And then finally, just to show that we actually saw increases in sensitivity with some of the compounds with the primaries. This is showing lead. So lead, of course, this is a known developmental neurotoxicant, and only the cerebellar granular cells at the higher concentrations showed a decrease. So what this showed us was that to increase the sensitivity to the screen, it's really valuable to use multiple models to cast as broad of a net as possible. One of the advantages of using high content is because the technology is automated, it gives the ability to look at multiple time points simultaneously. And what I wanted to share here briefly, this is showing a new endpoint, so total branch points in blue, in addition to the neurite length and viability that we looked at previously. And I wanted to share this because, so these compounds are pyrethroid pesticides. Pyrethroids are used very regularly within agriculture, and there's some concern about their safety. So bifenthrin, when we look at bifenthrin at all dose responses, it's not until we get to the higher concentrations that we see a decrease in both total neurite length as well as branching. However, for other compounds like cypermethrin, another one of these pesticides, notice that only branching shows a change. Neurite outgrowth and viability are relatively unchanged at all concentrations tested. And what that shows us is that if we evaluate other endpoints, such as branching, we're able to increase the sensitivity of our assay so that we're not missing any compounds that may be causing toxicity. And then last thing, as I wrap up here, this is showing synaptogenesis. And synaptogenesis is an assay that can only be done in primary cells. What I want to show, just in the interest of time, is here we see synaptogenesis assay. So channel 1, nucleus, channel 2, the beta-3 tubulin, where we see the Neurite outgrowth, channel 3 in red, the synapse formation. And if we look at this, high content technology is great for this type of evaluation, where we can validate only the neurons. We can exclude the glia, like we see here, based upon size and shape. We can take the resulting neuron morphology and along the neurites. So here we see the tracing of the neurites that are being detected. That becomes our region of interest. And along that region of interest, we can identify the synapse formation. And what was really interesting to us is that synapse formation that we were looking at using high content technology matched methodologies that are much more resource and time required to conduct the assay. So in a much more efficient screening assay, such as high content, we can show that level of synaptogenesis, as we had showed here. And with that, in the interest of time, I'll go ahead and wrap it up and pass it on to my colleague, Michael Derr, who is a scientist within cell biology.
Thank you, Nick. So the title of my talk is Neuronal Applications of High Content Analysis, and I'm going to show some examples of assays that we have developed and frequently used throughout the media development process, and hopefully provide some insight into the assay development process. So just an overview of the examples I will go over. So there's cell population characterization, I guess more specifically neuron counting, and then multi-parametric high content analysis of neuronal maturation, and then finally glial cell quantitation in primary neurons. And just kind of before I get into the examples, I want to give a brief description of what high content analysis is. So it's basically automated image capture coupled with automated image analysis software, and the software allows quantitation of basically hundreds of parameters within a single field. And the instrument that we are using is the Cell Insight CX5, and that is coupled with the HGS Studio image analysis software. So kind of getting into the assay development process a little bit, I like to show this image as an example of a culture that's not really ideally suited for high content analysis. You can see it's a highly mixed population of really densely packed cells, and the cells are kind of on top of each other and forming mounds and kind of clumping. And these are culture characteristics that make it very difficult for the software to reliably identify objects and make accurate measurements on the images. So part of the assay development process involves trying to make the biology facilitate kind of the technology in a sense. So this is kind of an extreme example in this image, but some ways of doing this involve adjusting seeding densities or experiment endpoint times, or trying different reagents if possible. So along these lines, antibody selection is very important. Localization of your antibody, specificity, and like high signal to noise ratios are definitely things to consider. And yeah, as I previously described, the spatial distribution of the cells is also very important. Okay, so now I'm going to get into the first examples. I'm going to show an example of neuron counting, and this kind of illustrates why antibody selection is important. Here we have day 21 primary rat cortical neurons that are stained with 2 different neuron-specific antibodies. And the MAP2 is on the left, and then UCD in the middle. And you can see that the MAP2 has kind of a diffuse staining pattern. It stains both the neurites and the cell bodies, while the UCD antibody is localized to the cell bodies only. And in the third image, you can see that both of these antibodies are staining the same cells. They're both neuron specific, so it's important to kind of make sure that your antibody has the correct specificity. And so now I'm going to kind of show why the cell body staining antibodies simplify the neuron counting process. And this is a good example of how the biology can facilitate the software. So by using the cell body localized antibody, we can use a very simple image analysis assay called the target activation assay. And so yeah, this is a very simple assay that is also very versatile. We actually use this in many, many different applications. But so just kind of to show how this assay works, first you use the DAPI stained nuclei, here seen in blue on the left, and the software identifies objects in the DAPI channel. And if you can, hopefully you can see the yellow mask that's generated there. And so then the software kind of duplicates the DAPI mask for the UCD channel and measures the intensity for each of the identified objects. So the low intensity objects are your non-neuronal population and are not counted, and they're actually in red now in the right image, the low intensity ones. And then the high intensity objects are the UCD positive neuronal population, and they're still kind of in yellow. Sorry about the colors aren't ideal in this image, but so the output from this assay can be the number of neurons or the percent neurons. And again, cell body or even nuclear localized antibodies really allow for simplified neuron counting. And this is just a data representation of why the UCD antibody is good for counting. We see the cell intensity population distribution illustrated as a histogram here. And so for this, the target activation assay was run using either the MAP2 antibody or the UCD antibody. And on the bottom panel, the UCD object intensity distribution shows a very clear kind of resolution between the neuronal population, which is highlighted in green, and the non-neuronal population, which is highlighted in red. So on the x-axis is intensity, so the higher intensity population is on the left, and then closer to 0 you have the non-neuronal low intensity population. And so comparing that to the MAP2 histogram on the top, you can see that that histogram, there's a lot of overlap of the positive and negative population. So again, UCD enables clear resolution of the neuronal and the non-neuronal population. And here I'm just showing that the UCD antibody can also be used to quantify iPSC-derived NSE differentiated neurons. Whenever you're using antibodies for a system to identify a specific cell type, you definitely want to make sure that they are staining the correct cells. So here's shows that the UCD and the MAP2 are staining the same cells in these iPSC-derived neurons. Okay, so now moving on from the neuron quantitation, we're going to look at an example of quantitative analysis of neuronal maturation. And this is a great example of how the improved culture characteristics can facilitate the software and enable accurate quantitation. So this is a differentiation time course for iPSC-derived NSC differentiation to neurons with CultureOne. And so at day 0 of this experiment, NSCs were plated in differentiation media with CultureOne. And the cells were fixed at day 2, day 4, day 7, and day 14. And then they were stained with the MAP2 antibody. And so early on at day 2 and day 4, you can already see nice neurite outgrowth. And as time goes on, the cells continue to mature, forming these really nice complex neurite networks you can see in day 7 and certainly at day 14. And also the cells are really nicely spatially distributed, and there are virtually no progenitor cells present. The bottom panel shows the software-generated object identification mask for each corresponding image, and the cell bodies are seen in kind of the turquoise color, and the neurites are in purple. And you can see that the neurites are being accurately identified if you compare the images. So basically, because CultureOne eliminates the clumping and the progenitor cell proliferation, it really simplifies the object identification process and improves the accuracy, and it really decreases the assay development time because you're not having to really train the software very much to identify the objects. And here is the data output from the time course experiment. This is the multi-parametric analysis of neuronal maturation. And for this, there's 6 different parameters that I measured, and all of these are kind of metrics of maturation. So on the top left, you have the neuron count, or the QCD positive cells. And you can see as time goes on from day 2 to day 14, your neuronal count is increasing. And then below that one, you have the NSC count, which is just really around 2 to 3 cells per field. It's very low, and it does not increase over time, suggesting that you've gotten rid of the proliferation of the progenitors. And then the right 4 graphs have to do with neurite quantitation of different kind of aspects of neurites. So you have neurite length and neurite area on the top, and those are increasing over time. And then you have neurite branch points and neurite width. So all of these, with increasing neuronal maturation, you get increased complexity of your neurite network, and these are all measured here. So it's a nice way to kind of monitor maturation over time. So right, again, the enhanced maturation of CultureOne really facilitates reliable and data-rich analysis of your differentiated neurons. And this is a very similar assay setup to the drug screening or the toxicity assay that Nick was speaking to earlier, and it also is very similar to how drug screens are done. Okay, now so shifting gears, I'm going into the third example of astrocyte quantitation. So this is actually a slide from David Cunninger's talk that was mentioned earlier, and these are day 21 mouse cortical neurons, and we've developed a new application for CultureOne that allows you to suppress your glial cell population and also control your glial cell population. So on the top you have, day 21 mouse cortical neurons with no CultureOne treatment. And on the bottom you have CultureOne treatment starting at day 0, day 3, and day 7. And then these cultures were then cultured for 21 days. And we show that if you start CultureOne treatment at day 0, you get complete suppression of your astrocytes. And then if you delay the treatment to day 3 and day 7, you get increasing amounts of astrocytes. So you can really control the levels of your astrocytes. So how we did this quantitation is demonstrated here. So your astrocytes generally kind of grow in together in little areas, so it's important to do a large number of fields. So here's a montage of 16 fields, and on the right is the object identification mask generated by the software, and the readouts for this is total object intensity per well and object area per well. And here's just the same slide from before, but I substituted in the 16 field montage images so you could get a better understanding of what's going on over the entire surface of the well. And here is, it's kind of the same story for the oligodendrocytes. If you start CultureOne treatment at day 0, you can get complete suppression of your oligodendrocytes, and then if you delay the treatment, you get increasing amounts. And again, getting short on time, but this is just how we are identifying these. The mask is on the right. So to wrap up the conclusions here, high content analysis is a highly versatile tool for analysis of neuronal cultures. Initial assay development considerations, antibody selection, and culture characteristics can dramatically improve accuracy and simplify quantitation. And then CultureOne Supplement enhances the maturation of NSCs, yielding ideal culture characteristics for high content analysis. And CultureOne Supplement can be used to suppress and control the levels of glial cells and primary neurons. So that's it, and thank you for listening.
Thank you for that informative presentation. Before we get started on the question and answer session, I would like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. Our first question is for Dr. Beacham. Can you add CultureOne Supplement to cells at different times during the culture? What is the outcome?
Yeah, you sure can. Basically, the data that I showed was with CultureOne added on day 0, and then we followed the trajectory of the differentiation over 3 weeks. Adding at different time points will allow the progenitor cells to proliferate more, so you can kind of control or titrate the cell numbers by when you add it. I'll point out, though, that it doesn't act like an antimitotic. It again, basically arrests division and steers cells toward a differentiated phenotype more quickly. So, and also some of Michael's data also hinted at this, showing different time points that you can add it to primary neuronal cultures. I hope that answers your question.
Thank you, Dr. Beacham. This next question is also for you. Can flow dye be used in microplate assays, or is it used limited to high frame rate microscopy and imaging?
Yeah, flow membrane potential dye is a fast membrane potential indicator dye that is best used in microscopy. You need very high frame rates to measure action potentials, but it's been used to do so. That said, I've seen now a couple of papers in the cardiac safety space where researchers have monitored membrane voltage with FlowVolt in a microplate format while pacing cells or monitoring spontaneous beating, and looked at changes in interspike interval with drugs of interest. Cardiac myocytes have a much slower sort of kinetics than the neurons do, and so relatively slow frame rates can resolve it. But by and large, its use is in high frame rate acquisition microscopy.
Our next question is for Dr. Radio. What range of Z values are typical for HCA morphology measurements?
Typically, you see between 0.3 and 0.4 Z prime values, and that is relative to the complexity of just the neurite outgrowth biology that occurs in the cells. There have been some assays more recently, and actually a pretty complicated model, a primary hippocampal model, where they published a Z prime value of a 0.73. That was out of Vance Lemon's lab at the University of Miami. I would say that that's a extremely good Z prime value given the complexity of the assay. But as far as a range, anywhere from 0.3 to 0.7 is what has been noted in the field.
Thank you. Our next question is for Michael Derr. Can CultureOne be used to suppress glial cells in a mouse hippocampal neurons?
It sounds like Michael might have some trouble tuning in. If we don't get an answer here in a second, I can speak to that. Okay, well, yes, the short answer is yes. It can be used on primary neural cultures where, again, as his data indicated, it will suppress the proliferation of glial and immature cell types or dividing cell types. So I showed a bunch of data with CultureOne in the context of neural stem cell derived cultures, and then Michael followed with a fair bit of data also looking at primary cultures where glial and other dividing cells can contaminate and overrun the culture. And so yes, it will suppress glial proliferation in primary cultures.
Thank you. The next question is for Dr. Radio. What other endpoints aside from neurite outgrowth can be informative for HCA screens?
Yeah, that's a good question. So in addition to the neurite outgrowth, other measurements that are known to be valuable are branching, like what we showed in the presentation there with the pesticide model. That's a good secondary endpoint that has different targets than what we see for neurite outgrowth formation, and as a consequence, it can show different sensitivities to toxicant exposure. More recently, people are turning to things like pre- and postsynaptic puncta to evaluate neurotransmission, and both of those things are known to be critical for the development of the healthy nervous system, so I'd say those are good targets as well.
The next question is also for Dr. Radio. On slide 45, the cytotoxicity was measured over time, or was it measured at only one time point? How does it change over time?
That's a great question. Yeah, so when we were initially setting up the assay, we did it as a time course where we looked at multiple time points. We ended up selecting 96 hours for our screen, which is the data that I showed. And that was just because of the screen and the number of compounds and the number of concentrations. We did that at one fixed time point. But leading up to it for the optimization work, it was looked at across hours as well as days. I hope that answers your question.
Thank you. The next question is for Dr. Radio. Can the readouts be combined with probes for mitochondrial function in HCA platform, given that mitochondrial function and health is considered important for neurite outgrowth or synaptogenesis?
Again, that's another really good question by the audience. So yeah, you can evaluate it. What we would do is multiplex it in a different channel outside of the Neurite outgrowth measurements that we were looking at. And what you can do is you can do something like a mita tracker, and that would allow us to look at the mitochondrial number. You could look at the morphology of the mitochondria, and based upon the intensity of the dye, you can get a sense for the membrane polarity. And all of those things can be simultaneously measured using the neuronal profiling application that's available on our high content technology.
The next question is also for Dr. Radio. What is the biggest challenge encountered by researchers when using HCA for neurobiology applications? And do you have any suggestions for overcoming this?
I think the biggest challenge remains the extrapolation step. When we talk about simple neuronal cell lines, like the PC12s and NS1 cells in my presentation, there's a lot of concern about how well those more simple cell models extrapolate into in vivo conditions. It's much more challenging to work with primary cells just from the need to have an animal facility potentially, the additional culture work. In some primary models, they don't like antibiotics, so contamination is more easy. And because of those complexities, it's more difficult for researchers to do. I think it's why some of the topics that we're talking about with the media, as well as some of the new technologies that are available on the imaging side, such as integrated confocal, are really important so that the scientists can be enabled to look at those more complex biologies and more complicated cell models.
Thank you. Okay, it looks like we have time for one last question. Our next question is for Dr. Beacham. Neuronal activity indicating dyes have typically had issues of toxicity. How do the flow volt and potassium indicators fare?
Sorry, I didn't quite catch the caveat there. So indicator dyes have a problem with what? Could you repeat the question?
Yes. Neuronal activity indicating dyes have typically had issues of toxicity. How do the flow volt and potassium indicators fare?
So I'm interpreting this question as, are these dyes toxic? The short answer is no, not in the time course of their utility. Flow volt dye acts just like Flow4 or other calcium indicator dyes. It's loaded into cells as a Fluxor dye, rather, is loaded into cells as an AM ester, where it resides for something on a time course of an hour to 90 minutes, but can certainly stay a good deal longer in the context of a high throughput screen. Cells respond with the same fidelity at 1 hour and 4 hours and 8 hours after loading. Flow volt as well. I'll point out loading with this dye is sort of cell type specific. Typically, loading is very short with flow volt membrane dye. You apply it for 15 to 30 minutes and then remove it. Overloading the dye will blunt the response, but is not toxic to the cells. I hope that answers the question. I'm not sure if I quite caught what the real question was.
I would like to once again thank Dr. Daniel Beacham, Dr. Nicholas Radio, and Michael Derr for their presentations. I would also like to thank our sponsor, Thermo Fisher Scientific Gibco, for making today's educational webcast possible. Today's webcast will be available for on-demand viewing through January 2018. You will receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. See you next time, and goodbye.